Hello everyone, welcome to the channel. Hope you all are well and fine in tackling the pandemic with renewed energy. I am Anandita Buragohain, research scholar from the Department of Zoology, Cotton University, under the guideship of Dr. Anjana Singha Neram. This is a part of the online educational resource program taken up by the Zoological Society of Assam. So, I would like to take this opportunity to thank the society for coming up with this initiative. This presentation is on the topic protein sorting and mechanisms of vesicular transport following the Zoology UG second semester syllabus of Cotton University. It is thereby aimed for the undergraduate students studying in different colleges and universities. Now, starting with the topic, let us first have a look into the learning outcomes of this presentation. The learning outcomes will be: what is protein sorting, how protein sorting takes place in the cell, the different processes of protein movement, what is vesicular transport, how transport vesicles function, and the different types of vesicular transport. Before going through the topic elaborately, let us first have a look into the historical background. In 1971, Gunter Blobel along with David D. Sabatini conducted experiments on the translocation of proteins across membranes and proposed the signal hypothesis. Gunter Blobel was awarded the Nobel Prize in 1999 for his discovery that many proteins have a signal sequence, that is, a short sequence of amino acid at one end of the protein which functions as a postal code for the organelle that is targeted. On similar lines, in 1973, G.E. Blair and R.J. Ellis proposed the envelope carrier hypothesis, which involves the movement of proteins from cytosol into chloroplast. In 2013, the Nobel Prize in Physiology was shared by J. Rothman, R. Skakman, and T. Sudoff for their roles in elucidating the makeup and function of cell vesicles. Before going into the details of how proteins are sorted inside the cell, it is important to know how proteins are synthesized. Proteins are synthesized by ribosomes. As the DNA in the nucleus of the cell is transcribed to mRNA, this mRNA moves out into the cytosol through nuclear pore and gets translated into proteins with the help of ribosomes. Translation of proteins, therefore, may occur in free ribosomes present in the cytosol as well as in membrane-bound ribosomes of the endoplasmic reticulum. Now comes the question, what is protein sorting? Protein sorting or protein targeting is the biological mechanism by which proteins are transported to appropriate destinations in the cell, such as the nucleus, mitochondria, chloroplast in plant cells, endoplasmic reticulum, peroxisomes, etc., or outside it, such as in the case of secretory proteins. Thus, proteins can be targeted to the inner space of an organelle, different intracellular membranes, plasma membrane, or to the exterior of the cell via secretion. This process of delivery of protein can be carried out with the help of information contained in the protein itself. It is important for proteins to be sorted in a correct manner, reaching the correct target destination. Errors in protein sorting can lead to various diseases such as adrenoleukodystrophy, which is an X-linked disease caused due to the abnormal functioning of enzymes resulting in fatty acid buildup. Protein sorting by free and membrane-bound ribosomes. As we know that proteins are translated by free and membrane-bound ribosomes, the proteins formed as a result of translation are sorted differently. In the case of free ribosomes, proteins formed are targeted into the nucleus, mitochondria, chloroplast, and peroxisomes. On the other hand, proteins that are translated by membrane-bound ribosomes are mainly secretory proteins which are targeted to the secretory vesicles, or they can be targeted to the endosomes and then to the lysosomes, or they can be translated as membrane proteins which are targeted to the plasma membrane. Protein movement is reliant upon three well-defined processes. The first is gated transport, which occurs between the cytosol and the nucleus. This type of transport takes place through the nuclear pore complex. The second is transmembrane transport, which is the transport of proteins from the cytosol to organelles such as the mitochondria, endoplasmic reticulum, peroxisomes, and plastids in plant cells. This type of transport is carried out through the protein translocators. Protein movement also occurs through vesicular transport, that is, from one organelle to another, and through transport vesicles. The figure shows the different processes, that is, the gated transport between cytosol and nucleus in red, the transmembrane transport from the cytosol to different membrane-bound organelles in blue, and the vesicular transport from the endoplasmic reticulum to different organelles, as well as from the cell surface to lysosomes in green. Let us start with the first process of protein movement, gated protein transport. This type of protein transport occurs between the cytosol and the nucleus, as we have already learned. Molecules which are smaller in size, that is up to 50 kilodalton in size, enters the nucleus by passive diffusion normally. However, molecules which are greater than 50 kilodalton, as in the case of proteins, cannot enter the nucleus passively. So such molecules need to be actively transported into and out of the cell. So, how does the nucleus import and export such proteins? Protein transport to the nucleus. Proteins destined to be imported or exported from the nucleus require a specific amino acid sequence. In the case of protein import to the nucleus, proteins such as histone and DNA or RNA polymerases contain nuclear localization signals NLS, which is the specific amino acid sequence required for protein import. The protein with the NLS moves post-translationally and after it is completely folded. Another protein, importin, binds with the NLS of the protein to be imported, also called the cargo protein. The importin cargo complex then translocates through the nuclear pore complex by interaction of the nuclear pore complex with importin. After reaching the nucleoplasm, the importin interacts with REN-GTP, which is a monomeric G protein. This interaction causes a conformational change in importin, leading to its dissociation from NLS and releasing the cargo protein into the nucleoplasm. The importin REN-GTP complex now diffuses back to the cytoplasm through the nuclear pore complex, and importin is thus freed into the cytoplasm. Similarly, nucleic export of proteins such as tRNAs, ribosomal subunits, and mRNAs require a signal sequence called the nuclear export signal or NES that stimulates the export from the nucleus to the cytoplasm. Another nuclear protein, exportin, binds to the NES of the cargo protein. REN-GTP, which is present in a nucleoplasm, simultaneously interacts with exportin, resulting in an increased affinity of exportin for the NES. This leads to the formation of a trimolecular cargo complex, which translocates out of the nucleus through the nuclear pore complex. In the cytoplasm, REN hydrolyzes the bound GTP, leading to the release of exportin and the protein cargo into the cytoplasm. Thus, protein gets exported out of the nucleus in this way. The second process is the transmembrane transport. Taking the example of transmembrane translocation of protein in the endoplasmic reticulum, protein translocation in ER occurs through co-translational and post-translational mechanisms. Protein translocation through a biological membrane such as the ER membrane requires a signal sequence, which is a small sequence of amino acid that helps in targeting the protein through a specific translocator. So let us first know what a signal sequence is. Signal sequences act as postal addresses on the protein that helps them in reaching their target destination. It is a short amino acid sequence known as signal peptide or signal sequence. It contains specific targeting or sorting signals that directs their transport from the cytosol to specific organelles of the cell. Proteins that do not contain sorting signals remain as permanent residents in the cytosol. The picture shows the figure of a standard signal peptide. It contains an N-terminal sequence that helps in targeting the secretory and membrane proteins into the ER. These have a tripartite structure having a hydrophobic core region flanked by an N and C regions. The C region contains the site for cleavage by signal peptidase. Usually, these are cleaved off co-translationally to produce a mature protein and a cleaved signal peptide. Before going into the details of how protein is imported into the endoplasmic reticulum, let us first learn what signal hypothesis is, which was first proposed by Blobel and Sabatini in 1971. This hypothesis included the work of over 2 decades by Blobel and his co-workers. According to the signal hypothesis, proteins destined to be secreted from cells are synthesized initially with 15 to 30 amino acids at the amino terminal of the protein. This peptide sequence initiates binding of the translation complex to the RER membrane. The result is the transfer of protein across the RER membrane to its lumen. The signal sequence, which is the 15 to 30 amino acid sequence, is cleaved by a specific protease signal peptidase. The hypothesis also stated that the signal sequence as it was synthesized was recognized by a protein, the signal recognition protein or SRP, in the cytosol which binds to the signal sequence. The SRP then binds to its receptor present in the ER membrane and helps the ribosome tether to the ER. We will look into the details in co-translational translocation in the coming slide. Now, let us have a look into the protein transport into the endoplasmic reticulum. Protein transport into the endoplasmic reticulum is of two types, co-translational and post-translational translocation. That is, proteins will be translocated co-translationally or it can also be translocated post-translationally. In co-translational translocation, as the mRNA is being translated to protein, a cytosolic protein called signal recognition protein SRP binds to the signal sequence of a nascent polypeptide chain. The complex formed by the SRP, nascent polypeptide and ribosome then bind to the alpha subunit of the SRP receptor in the ER membrane. The SRP receptor contains two subunits, a peripheral alpha subunit and a transmembrane beta subunit. The SRP, having two binding sites, directs both the binding of the ribosome to the ER membrane and insertion of the nascent polypeptide into the transmembrane channel. Thus, the polypeptide chain elongates and the signal sequence is cleaved by a signal peptidase. In post-translational translocation, completely synthesized polypeptide is maintained in a competent state by cytosolic chaperone proteins and targeted to the post-translocone in the ER membrane. It consists of the Sec61 translocone and the Sec62-63 complex. The J domain of Sec63 recruits and activates the luminal BIP, which acts as a molecular ratchet and facilitates the translocation of the polypeptide chain. After cleavage of the signal sequence by a signal peptidase, the polypeptide chain is released into the ER lumen. In both co-translational and post-translational translocation, a signal peptidase cleaves the signal sequence soon after N-terminus enters the lumen. Now comes the question, what happens to a protein after it enters the ER lumen? After the protein enters the ER lumen, certain modification events take place. One of the major events is glycosylation. Glycosylation in simple terms is the addition of one or more carbohydrate to a protein, resulting in the formation of a glycoprotein. A preformed N-linked oligosaccharide precursor is added to the protein in the endoplasmic reticulum. The precursor oligosaccharide is assembled on a membrane-attached anchor called the dolichol phosphate, a polyisoprenoid lipid. A branched oligosaccharide containing 3-glucose, 9-mannose and 2-N-acetyl-glucosamine molecules are added step by step to the dolichol phosphate, forming a total of 14 residue oligosaccharide. The entire 14 residue precursor oligosaccharide is transferred to an asparagine residue from the dolichol phosphate on a nascent polypeptide chain in the ER lumen. Glycosylation also continues in the Golgi complex, where mainly two classes of oligosaccharides are formed, high mannose oligosaccharide and complex oligosaccharide. In high mannose oligosaccharide, apart from the 2-N-acetyl-glucosamine, many mannose residues are added. On the other hand, in complex oligosaccharide, galactose and sialic acid residues are added in variable numbers. Glycosylation is important in that it helps in the folding and provides stability to the glycoproteins. Protein folding in ER and movement through the secretory pathway. After glycosylation of the polypeptide, 2 of the 3-terminal glucose is removed by the enzyme glucosidase. The presence of terminal glucose allows the binding of the polypeptide to several chaperone and ER proteins that helps in proper folding and renders stability. Once folded properly, the terminal glucose is removed and the protein becomes ready to get transported to the Golgi. However, misfolded proteins either remain in the ER or are degraded by proteasomes. The correctly folded proteins exit from the ER to the cis phase of the Golgi complex and undergoes further modification and gets released from the Golgi through the trans phase in secretory vesicles to target destinations. If certain proteins that were supposed to remain in the ER somehow gets translocated to the cis Golgi, can be retrieved back to the ER. This is because these ER resident proteins contain retrieval signals. The vesicles transporting them as well as the cis Golgi network have receptors specific for these retrieval signals that help in the retrieval of ER resident protein. Soluble proteins called KDEL sequence, that is lysine, asparagine, glutamine and leucine amino acid, acts as a signal for retrieval. And membrane proteins contain KKXX sequence, that is lysine, lysine XX, where X can be any other amino acid, which acts as a retrieval signal. Envelope carrier hypothesis. The envelope carrier hypothesis was proposed by Blair and Alice in 1973. According to the hypothesis, although chloroplasts synthesize small portions of their required proteins, the rest are synthesized in the cytosol and then imported into the chloroplast. So earlier it was thought that chloroplast as well as mitochondria synthesize the required set of proteins. However, although small portions such as the large subunit of Rubisco enzyme, for example, is synthesized in the chloroplast, later it was found that the small subunit is synthesized in the cytosol and then transported to the chloroplast. It also proposes that proteins destined for chloroplast and synthesized in the cytosol require a recognition event for their unidirectional entry into the organelle. So, these proteins which move from the cytosol to chloroplast also require a signal sequence for their entry through the chloroplast membrane. Later, this hypothesis was found to be true for mitochondrial protein transport as well. So, Now let us have a look upon the transmembrane protein transport to mitochondria and how it takes place in the cell. Protein transport to mitochondria. Mitochondria is a double membrane bound structure having an outer and an inner membrane. The inner membrane is highly convoluted, forming cristae. Mitochondrial proteins which translocate from the cytosol to mitochondria contain mitochondrial targeting signal sequences. The cleavable ones, the cleavable signal sequences, are called pre-sequences. These are rich in positively charged basic amino acids such as arginine and lysine. The proteins which are synthesized by cytosolic ribosomes are maintained in an unfolded state by cytosolic chaperone HSC70. The pre-sequences of the proteins first bind with the translocase of outer membrane complex or TOM complex, which directs and transfers the protein through the translocone present in the outer mitochondrial membrane. The pre-sequence directs protein through the translocase of outer membrane complex or TOM complex to the translocase of inner membrane complex or TIM complex. After the pre-sequence of the protein enters the matrix, it is removed by mitochondrial processing peptidase MPP, and the imported protein is then folded by the mitochondrial chaperone proteins. Proteins are also targeted to peroxisomes. So, now let us have a look into the protein transport to peroxisomes. Peroxisomes are small organelles bound by a single membrane containing oxidative enzymes. Peroxisomal proteins are translated in free ribosomes in the cytosol and are fully folded before their transport to peroxisomes. Proteins that are imported into peroxisomes contain peroxisomal targeting sequences PTS. PTS in peroxisomal transport proteins are bound by PTS receptors. Here in this case, PEX5 receptor acts as the PTS receptor and it binds with the PTS in the cytosol itself. It then interacts with the PEX14, which is a part of the translocone responsible for the import of the protein. This interaction leads to the import of the protein into the peroxisome and releases the receptor back into the cytosol. The third process of protein movement is vesicular protein transport. Vesicular transport is the predominant mechanism for exchange of proteins between membrane-bound organelles in eukaryotic cells. Transport vesicles are bubble-like membranous structures that stores and transports cellular products. It forms an intracellular membranous sac that is separated from the cytosol by at least one lipid bilayer. Transport vesicles play a central role in the traffic of molecules between different membrane-enclosed compartments of the secretory pathway. That is, transport vesicles are needed in the secretory pathway for the transfer of secretory proteins. Vesicles are similarly involved in the transport of materials taken up at the cell surface. As we can see, vesicles budding off from the ER rich and fused with the cis-Golgi network and vesicles budding off from the trans-Golgi network fusing with the plasma membrane and subsequently secreting their contents to the outside. Vesicular transport is thus a major cellular activity responsible for molecular traffic between a variety of specific membrane-enclosed compartments. Having known what is vesicular transport, now let us have a look into how vesicles are formed and how they fuse with their target membranes. The fusion of a transport vesicle with its target involves two types of events. First, the transport vesicle must specifically recognize the correct target membrane, and second, the fusion of the two. Unique pairs of transmembrane proteins help in specific recognition between a vesicle and its target, followed by the fusion of the two bilayers. Rotman and his colleagues proposed a model called the snare hypothesis, according to which vesicle fusion with the target is mediated by interactions between specific pairs of proteins called snares, short for SNAP receptors, on the vesicle and target membranes called V and T snares respectively. All snare proteins contain conserved haptid repeat sequences. From the figure, as we can see that the transmembrane cargo protein binds with the protein. This leads to the initiation of vesicle formation, leading to the recruitment of coat proteins. The vesicle along with the cargo, its receptor and the V snare starts budding from the parent membrane. At this stage, accumulation of coat protein from the cytosol continues. The vesicle finally separates from the parent membrane and starts moving towards the target membrane. As it moves and tethers with the target membrane with the help of the tethering factor, uncoating of coat proteins occur which goes back to the cytosol. The active Rep protein binds with the tethering factor and the vesicle to allow the snare proteins, that is the V snare of vesicle membrane and T snare of target membrane, to dock. The binding causes the vesicles to remain bound for long enough to allow the Rep protein to hydrolyze its bound GTP, which locks the vesicles onto the target membrane. After fusion of the two membranes, the snare proteins separate and the protein cargo is transported to the target organelle. Different types of coat proteins on transport vesicles. Transport vesicles arise from specialized coated regions of the membranes which are surrounded by a coat of proteins covering the cytosolic phase so that these membranes eventually bud off as coated vesicles. Prior to fusing with the target membrane, this protein coat is discarded, as we have seen in the previous slide, to allow the membranes to fuse directly. There are mainly three types of coat proteins that cover the vesicles. These coat proteins are specific to the type of vesicles they bind with. It is seen that along with coat proteins, various adapter proteins and small GTP binding proteins are also needed for coated vesicle formation. The first type is clathrin coated vesicles, which form multiple complexes with different adapter proteins. Clathrin coated with adapter proteins 1 and 3 form vesicles from the trans-Golgi network to the lysosomes, and clathrins coated with adapter proteins 2 form vesicles from the plasma membrane to early endosomes. The second type of coat protein is COP1. COP1 proteins form vesicles for intra-Golgi transport and for retrograde transport from the cis-Golgi network to the endoplasmic reticulum. The third type is COP2 proteins. COP2 proteins carry out anterograde transport from the endoplasmic reticulum to the Golgi complex. Protein transport to lysosomes. Proteins destined for lysosomes are sorted by the presence of mannose-6-phosphate, which binds to mannose-6-phosphate receptors or M6P receptors present in the trans-Golgi. Binding of the protein to its receptor eventually leads to the budding off of vesicles from the trans-Golgi network, which then fuse with the lysosomes. This results in the releasing of the contents from the receptor to the lysosome, and the receptor eventually buds off from the lysosome and fuses again with the trans-Golgi network. Let us now have a look into the overview of vesicular transport, that is, the overall aspects of vesicular transport. In vesicular transport, substances are transported with the help of vesicles. Transport of substances, as we have seen, takes place across the membrane but not through it. The substances carried through vesicular transport cannot pass through membranes. Thus, it is a mode of transport for macromolecules. Transport of vesicles require metabolic energy. It is a type of active transport. And lastly, transport occurs independent to the concentration gradient. Types of vesicular transport. There are various types of vesicular transport. What we have learned so far is mainly related to the transport of proteins. As a whole, there are three types of vesicular transport. Exocytosis, consisting of the secretory pathway, about which we learnt in the previous slides. Secretory pathway can be of two types, constitutive and regulated. In the constitutive or continuous secretory pathway, proteins are continuously secreted. Examples are in the case of collagen by fibroblast, serum proteins by hepatocytes, and antibodies released by B lymphocytes. On the other hand, cells of the regulated secretory pathway secrete products on demand, that is, in response to specific signals. Cells of this pathway release their contents by exocytosis, as we already know. Example includes the release of hormones, neurotransmitters, and digestive enzymes. The second type is endocytosis, that is the movement of substances from the outside to the inside of the cell by vesicle formation. It is of three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. The third type of vesicular transport is transcytosis, where substances enter a cell by vesicle formation and move through it. Coming to the first type of vesicular transport, exocytosis. Since we have already learnt about the constitutive secretory pathway of proteins in vesicular protein sorting, let us look into the regulated secretory pathway, taking the example of release of neurotransmitters from the axon terminal to the next neuron. Neurotransmitters are present in synaptic vesicles. When the threshold action potential reaches the axon terminal, this forms a signal for the opening of calcium channels, resulting in the inward flow of calcium ions. This causes the vesicles to release the neurotransmitters into the synaptic cleft by fusion with the neurolemma. After its release, the transmitter binds to a receptor on the subsequent neuron, resulting in the activation of the neuron and generation of another action potential. The neurotransmitters released are inactivated and removed from the synaptic cleft by special transporter proteins on the presynaptic membrane, which carry the protein into the presynaptic neuron and repackage them into vesicles, or they are stored until needed again. Not all neurotransmitters are recycled back to the presynaptic neuron. Some neurotransmitters are degraded by degradation enzymes in the synaptic cleft itself. The second type of vesicular transport is endocytosis. Endocytosis is the cellular process in which substances are transported into the cell. The material to be internalized is surrounded by an area of the cell membrane, which then buds off inside the cell to form a vesicle containing the ingested material. The figure shows the formation of a vesicle by cell membrane. You can see how the membrane forms a ruffle and the non-cytoplasmic ends fuse to form the vesicle, which eventually pinches off. We have learned that there are three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is the engulfment of extracellular particles by the cell, example in the case of macrophages. Pinocytosis is the uptake of liquid matter by vesicle formation. This process is also known as cell drinking. Receptor-mediated endocytosis. Here, various enzymes or other substances bind with the extracellular receptors on the cell and are then taken up by vesicle formation. Example in the case of binding of LDL to its receptor and their uptake. Now, let us take a close look at the functioning of receptor-mediated endocytosis. Receptor-mediated endocytosis requires the vesicles to be coated with clathrin. Receptor-mediated endocytosis of low-density lipoprotein, or LDL, occurs through clathrin AP2 coated vesicles. Michael Brown and Joseph Goldstein demonstrated the uptake of LDL by mammalian cells. This requires the binding of LDL to specific cell surface receptors, that is LDL receptors concentrated in clathrin-coated pits. When a cell needs cholesterol, it synthesizes the transmembrane receptor proteins LDL receptor and inserts them into the plasma membrane. When the LDL binds to its receptor and are rapidly internalized in clathrin-coated vesicles. These vesicles then move to endosomes. The contents derived by early endosomes are directed to fuse with and deliver their membrane and content to late endosomes and then to the lysosomes. In the late endosomes, LDL dissociates from its receptor and the receptor pinches off from the late endosome to fuse with the plasma membrane facing the exoplasmic site for further binding. The LDL in the lysosome undergoes proteolysis and recycling in the lysosome's acidic environment to form different types of amino acid, cholesterol and fatty acid, which is then released into the cytoplasm. The third type of vesicular transport is transcytosis. Transcytosis is the transport of macromolecules across a cell. In this process, substances taken up by a cell move through it and get released out from the other side. It can be receptor-mediated transcytosis or absorptive transcytosis. In receptor-mediated transcytosis, substances are bound to extracellular receptors and taken up by vesicles. These vesicles move across the cell to fuse on the other side and release its contents. In adsorptive transcytosis, cationic molecules are adsorbed on the cell surface and taken up to the luminal surface of the endothelial cells. These move through the vesicles and moves out by exocytosis to the brain parenchyma. This forms a route for the delivery of medicinal drugs to the brain across the blood-brain barrier. These are the suggested readings and references. With this, I would like to wind up the video. Thank you for your patience and also please drop in your feedback and queries in the given handle.